Welcome to the Wright Science Daily Revision Boost. In today's Biology Blast, we're going to look at mutation and the history of genetics. First thing we need to know is that a mutation is the change in the base sequence of the DNA. Now, mutations can occur spontaneously as a result of DNA replication, or we could have an increased chance of these mutations occurring due to ionizing radiation like UV and certain chemicals like benzene and ethanol. When we're talking about genetic variation, this is different versions of an allele that arise from mutations changing the DNA. Now, mutations may show no effect on the phenotype, influence the phenotype, or determine the phenotype, which leads to variation within a species. When we get a mutation, it could either be harmful, beneficial, or neutral. If we consider harmful mutations first of all, they may cause cancer, where the cells grow and divide in an uncontrolled manner. They could cause abnormal channel protein production, like in cystic fibrosis, or they could cause different shaped protein molecules to be made, like in sickle cell anemia. Neutral mutations are things like tongue rolling or having free or attached earlobes. They have no impact on your existence whatsoever, just a fun quirk. And finally, the beneficial mutations. This could be antibiotic resistance in bacteria, or the FOXP2 gene, which gave us the ability to speak, or skin colour mutations. Hopefully those of you on the separate science higher tier know that DNA is organised into coding sections, which are the genes, which are separated by non-coding DNA. So if the mutation happens in the non-coding section, there's no effect, but if it happens in the gene, we could see the effects. So when a mutation happens in a gene, the DNA bases may be changed, added, or deleted. If we change a base, then that changes the one amino acid that's coded for by the sequence that's been changed. If we add a base, then this has much greater impact because it's going to change the amino acid coded for by that triplet sequence and all triplets that follow it. And if we delete a base, it changes the amino acid coded for by that sequence and then all the triplet sequences that follow it as well. So changing a base only affects one amino acid, but adding or deleting affects many more. If we've changed the amino acid sequence, then the way the protein folds could be changed, or we can actually just make the completely wrong protein. So do remember this links back into our work that we did on enzymes, transcription and translation from B1. What we also have are start codons, which are found before each gene in the non-coding section of DNA, and that triggers transcription. So if the mutation occurs in the start codon, then the gene may not be transcribed at all, which means the protein will not be made full stop. Another thing we need to know for the higher GCSE biology are the history of genetic scientists. So the first one, Gregor Mendel, actually carried out experiments with peas back in 1866. He observed characteristics were passed on from parents to offspring. So he noticed that characteristics in plants were determined by what he called hereditary units, and those units were passed on, one from each parent. And the hereditary units are either dominant or recessive. Our second scientist is Friedrich Miescher, who discovered nuclein. He discovered this acidic substance present in the nucleus. Scientist number 3 is Oswald Avery in 1944, and he transferred DNA between bacteria. He transferred genes. The ability to cause disease was then passed from one strain of bacteria to another, and those bacteria then passed on the ability to their offspring. So he showed that genes were made of DNA. Our next scientist is Erwin Chargaff, who discovered base pairs. So even though different organisms have different amounts of DNA, all DNA has equal quantities of adenine and thymine and equal quantities of cytosine and guanine. So a common question they like to ask you here is they'll give you some of the combinations and ask you to work out the missing one. So remember, adenine and thymine will be the same, cytosine and guanine will be the same. Next scientists were Rosalind Franklin and Maurice Wilkins. And Rosalind Franklin photographed DNA crystals using X-ray crystallography to produce these images of the DNA crystals, which were then used by James Watson and Francis Crick to actually identify the double helix structure of DNA. So they described it as the double helix, those two spirals held together by the complementary base pairs. Since our discovery of DNA was concluded by Watson and Crick, then we've actually gone on to discover individual genes that code for the inherited disorders like cystic fibrosis. We've also seen the development of the field of genetic engineering. By the time we get to 2003, then the Human Genome Project was completed, which was a 20-year project to sequence 24,000 genes, which are all of the genes in the human body. And since 2003 and the completion of the Human Genome Project, we've been trying to carry out gene therapy, which is where we're replacing faulty genes with normal copies. Don't forget, there are different ways that you can remember those scientists to help you out rather than just a string of names. So using little mnemonics like this might help you remember the sequence of the scientists. Today's chemistry check is all about titrations. So first of all, when we're talking about a titration, this is a method we use to determine the concentration of an acid or alkali by using a neutralization reaction. So we'll be using a single indicator like phenolphthalein as it gives a clear endpoint. So titration is one of our assessed practicals. Quick reminder on how we do this then. We fill the burette with acid, which is the long glass tube with a tap at the bottom. You use a volumetric pipette to transfer 25 centimeters cubed of your alkali to the conical flask. Add a few drops of the single indicator to the flask. Record your start volume from the burette and then gradually add the acid from the burette to the flask until it just changes colour, recording the end volume from the burette. Couple of points to note about ensuring accuracy. Number one, make sure the tap of the burette is closed before you start to fill it. Make sure that you're at eye level with the bottom of the meniscus so that you're getting accurate readings from there. And then as you approach the end point, remember to add slowly a drop at a time to get the most accurate reading. Another one that we can do is to make sure the flask is swirled or constantly mixed so that it gives that clear end point rather than it changes and then you give it a mix and it's not actually changed at all. What we should do is a rough titration first of all to give us an idea of where the end point is, and then we need to get concordant titers. So this means that they're within 0.1 centimeters cubed of each other. So a titer is just a measured volume of solution added from a burette during titration to reach the end point. In terms of the choice of indicators, there are three that are known as single indicators. So litmus, which is red in acidic and blue in alkaline. Phenolphthalein is colourless in acidic solutions but pink in alkaline, and methyl orange is red in acidic and yellow in alkaline solutions. We use these because there's a distinct colour change at the end point, so that means that you can clearly see when that reaction has ended, whereas universal indicator you're constantly trying to guess, therefore we don't use it. What we also need to do with our titration is have a standard solution. So a standard solution has a known accurate concentration that we use in titrations. In order to make it, we've got to measure a known mass of our actual alkali and dissolve it in a specific volume of water, and then we use a volumetric flask to top it up to our 250 or 500 centimeters cubed, whatever we wanted. So that means that the actual concentration of that solution in the flask is exact and known to us because we've made it very precisely. In terms of the equipment choice, then we could use measuring cylinders to measure our 25 centimeters cubed for the conical flask, but that's obviously way less accurate than a volumetric pipette. So if they give you a method where someone's used a measuring cylinder and asked for an improvement, use a volumetric pipette is fantastic. We also need to make sure that the burettes are clamped vertically, because if it's on a slight angle, then obviously that's going to give us inaccurate results as well. Just a couple of other points on the accuracy. As we've already said, read from the bottom of the meniscus with your eye level with that line. Record it to 2 decimal places. So if the bottom of the meniscus is on a line, then it's a 0, whereas if the bottom of the meniscus is between 2 lines, the second decimal place is a 5. You should always swirl the flask to mix the contents and add it drop by drop as we approach the end point. Once we've carried out the titration, then we know the two reactants, the volume and concentration of one of those reactants, and the volume of the other. And using that information, we can then calculate the unknown concentration. In order to do this, we need to remember this calculation. So the amount in moles is the concentration in mole per dm cubed times the volume in decimeters cubed. So if you know this triangle, then you're going to be able to do titration calculations. We've got 4 key steps to doing our titration calculation. First one, convert the volumes to decimeters cubed by dividing by 1000 if they're given in centimeters cubed. Secondly, calculate the amount of the reactant you know the volume and concentration for just by doing your calculation of your concentration times the volume. Then use the balanced equation to work out the amount of the other reactant. So look at the ratios and multiply accordingly. And then we can use our equation to work out the unknown concentration because we will have the number of moles and the volume. So it's just your number of moles divided by volume. Last bit of our chemistry check today is gas calculations. So 1 mole of any substance in the gas state occupies the same volume at the same temperature and pressure. So this is known as the molar volume. And at room temperature and pressure, this is 24 dm cubed per mole. So again, a little equation to remember at the bottom, volume in decimeters cubed is the amount in moles times 24 dm cubed per mole. So when they give you 5 moles and ask you the volume it occupies, it would be 5 times 24 and so on. In our physics prep today, we're going to have a look at atoms, isotopes and radiation. Hopefully we remember from our earlier work, the nucleus of an atom is made up of protons and neutrons. And hopefully we learned all of these key facts about the proton, neutrons and electrons in our previous exam series. So the relative mass of the proton is 1 and its relative charge plus 1. Neutron, mass of 1, charge is 0. An electron, mass is 0.0005 and the charge minus 1. Because the charge on the nucleus depends on the number of protons present, it's a positive charge. So if there were 50 protons, it would have a charge of plus 50. We should also know the definition for isotope, which is an atom with the same atomic number but a different mass number because it's got the same number of protons and electrons but a different number of neutrons. So you've got 3 different isotopes of carbon given to you there, carbon 12, carbon 13 and carbon 14. Do remember our atomic number is the number of protons or the number of electrons, and the mass number is your number of protons plus the number of neutrons. So when you're given those two, you can work out how many protons, electrons and neutrons there are. What we find is that most atoms are stable, so they don't decay. But some atoms are unstable because they've got this unstable nucleus that will emit radiation as it undergoes this process of decay. Any material that emits radiation is known as radioactive. In the table, we've got our 4 types of radiation that have been summarized with their key facts. So we've got 3 particles, alpha, beta and neutron, and 1 electromagnetic wave, which is gamma. So make sure that you do know what each of them actually are in terms of their type, their symbols, what they are in terms of fast-moving electron, particle in the nucleus, nucleus for helium atom, etc., and the equation symbol on the right-hand column there. Do remember that where we've got the numbers there, the top number is the mass relative to a proton, and the bottom number is the charge. When we're looking at this process of decay, then we can find that neutrons decay to make a proton and an electron. When it comes to detecting radiation, we use a piece of equipment called a Geiger-Müller tube or Geiger counter, and what that actually does is it produces little clicks that we can hear, and those clicks are tiny currents produced when the radiation ionizes atoms of the gas that's present within the tube. This diagram is an important one to understand. So what we've got is a diagram showing the different penetrating powers of our 3 key types of ionizing radiation they will ask about. So we've got alpha, beta and gamma there. Alpha is the least penetrating, stopped by only a few sheets of paper. Beta will pass through the paper but be stopped by a few mils of aluminium, and gamma passes through paper and aluminium but will only be mostly stopped by a few centimetres lead or a few metres of concrete. The kind of question they do like to ask you is give you a table of different counts based on what it's passed through and ask you to identify which radiation you've got. Now, don't be thrown, because sometimes they'll have a combination of two of those types of radiation. So it might be alpha and beta, for example. So just watch out for that. Because alpha, beta and gamma have different penetrating powers, it does mean that we can identify them from what they're able to pass through. The radiation that's emitted by radioactive material is known as ionizing radiation. And ionizing radiation can remove electrons from atoms to produce positively charged ions. So in this table we've got a summary about the mass, the charge, the ionizing power and the range of alpha, beta and gamma. So make sure that you do know those key facts about those three types of radiation. In order to ionize an atom, energy has to be transferred to it. And alpha particles transfer more energy to the material they travel through, so that means that they've got a short range because they're transferring their energy to ionize the material and therefore won't go as far. The last bit we need to know are the different nuclear equations that show these types of decay. So in alpha decay, an alpha particle is emitted. The alpha particle is made of 2 protons and 2 neutrons, so that if they give you that equation in the second box with a couple of bits missing, all you've got to do is make sure that the bit on the right adds up to the numbers on the left, or vice versa. So if you're given 240 and 94 on the left, and they've missed out the couple of numbers from uranium, then we know that it's got to be 236 and 92, because 240 minus 4 is 236 and so forth. In beta decay, a neutron is going to decay to a proton and an electron. So what we end up with, our fast moving electron is given the symbol on the far right of the second box there. And what we'll see is the mass number stays the same because the total number of protons and neutrons remains the same. We've lost 1 neutron, but we've gained 1 proton, so it stays as 14 in this example. Whereas our atomic number goes from 6 to 7 because we've also created a proton. Third type of decay are gamma decay. Because gamma rays have no mass or charge, the final type of decay are neutron decay. This is where the decay of some nuclei leads to the production of large numbers of neutrons. So these neutrons are then emitted to make it stable. And a good example of this is helium-5. So you can see we start off with helium-5 and then we lose a neutron, so it goes down to helium-4 plus our neutron on the right hand side. Now we've had a quick recap, then head on over to socrative.com and test your knowledge on the topics from today. The room name you need to enter is Wright 2662. Good luck! As always, if there's any topics we've gone through in our daily booster that you need a little bit more work on, head on over to the relevant playlist to get some more detailed descriptions. So for biology it was B5, chemistry is in the C5 playlist, and physics for the combined science people is in P4, and for the separate science people it's the GCSE Physics P6 playlist.